जी अस्सलाम वालेकुम बेटा। कैन यू हियर मी? अच्छा जी, लास्ट डे वी डिस्कस the rate of enzyme action and the different ways of calculating uh, the rate of enzyme action. And today we'll discuss the different factors affecting the rate of enzyme action. Okay. In this case, we have two parts. One is an investigation, investigate the effect of different parts, different factors on the rate of rate of enzyme action. And the other is explain the effect of different factors on the rate of an enzyme action. Okay, so today we will discuss explaining the effect of different factors on the rate of enzyme action. So let's see factors affecting the rate of an enzyme action. So following factors affect the rate of an enzyme action. Number one, temperature. Number two, pH. Number three, substrate concentration. Number four, enzyme concentration. And number five, inhibitors. So we'll discuss them one by one. So let's see effect of temperature on the rate of an enzyme action. Effect of temperature on the rate of an enzyme action. Okay. So we will have three different types of questions on this topic. We will have questions beginning with describe. We will have questions beginning with an explain, and we will have questions beginning with investigate. Okay. So let's see if I draw the effect of temperature on the rate of an enzyme action on a graph. This graph has temperature on x-axis and it has rate on y-axis. Okay. So as this temperature increases, we can carry out an experiment at different temperatures, starting with 0, maybe 10, maybe 20, maybe 30, 40, 50, and maybe 60, and so on. At different temperatures, we carry out the enzyme activity and record or calculate the rate at different temperatures. At 0, the rate of enzyme action is 0. And as we keep on increasing the temperature, the rate of enzyme activity that increases up to 40 degrees Celsius. This rate increases up to 40 degrees Celsius. Okay. But after 40 degrees Celsius, the rate decreases, and then that ceases at around, maybe 50 degrees Celsius. So you may have two questions on this graph. Number one, maybe describe the effect of temperature on the rate of enzyme action. of an enzyme action. And the other will be explain the effect of. So how do these two answers differ from each other? We will discuss. So this begins with describe. In this case, what you should do is you should write the reference of temperature from an x-axis. Like when we increase the temperature from 0 degrees Celsius to 40 degrees Celsius, the rate increases from this. Maybe you will have numerical value here to this numerical value that you would find the numerical value here. So this is known as, the numerical code. So you should write increase of temperature from 0 degrees Celsius to 40 degrees Celsius increases the rate from, you can find these numerical values from here. Whatever the numerical values are there, you can write from A to B. From A with its units to B, whatever the numerical reference is there or numerical value is there, to B units. Okay. So this is, there are 2 points here. One is you are talking about, the relationship, increasing the temperature increases the rate. And second is the numerical code that you have taken from an x-axis and y-axis. So this is for 2 marks. Then you write here that the rate is maximum at 40 degrees Celsius. This is the second point that you can write. The rate is maximum at 40 degrees Celsius. And then you can write the rate slows down from 40 degrees Celsius to maybe above 40 degrees Celsius. Okay, so this rate slows down. And this is the max. And this is rate increasing. Okay, and then the enzyme activity that ceases at this temperature, you will have to find this temperature from x-axis, and you can write that enzyme activity ceases here. Or you can just say that rate of enzyme action is 0 at this point. Rate is 0 at this temperature. Why? Because that touches this x-axis. So this is how you should describe it. Okay. Then if this question is about explaining the effect, then what you should do? If the question is explain the effect of temperature on the rate of an enzyme action, then what you should do? Now, in this case, you need to give reasons for that. Okay. Explain why, or give reason. Give reasons for this increase in rate, and give reasons for this, the max rate here, and give reasons why this rate slows down here, and give reasons why this, this enzyme activity that, or the rate becomes 0 at this point. So let me just copy it here from here. And okay, just give me a minute. So here I'll insert this. So now if it has to be explained, then you have to give reasons why this rate increases here. You should give reason why this rate that slows down, and you should give reason why this enzyme activity that ceases at this point. So here, when you increase the rate from this point, from this A point, maybe to maybe the M point, or where the rate is maximum. Here, as the temperature increases, the kinetic energy increases. So more kinetic energy. If there is more kinetic energy, these enzyme molecules here, there are enzyme molecules. We can draw this enzyme molecule showing this, and the substrate molecules here. So these are the substrate molecules. So more kinetic energy, more collision in between these substrate molecules and these enzyme molecules would collide at a faster rate. Okay, so faster collision or higher the collision rate, or more collision between enzyme plus substrate, and so consequently more enzyme substrate complexes will be formed, and higher the rate. Okay, higher the rate. So that's how you should have these 4 points to explain why the rate increases if you increase the temperature. Okay, at this point, there is this at this temperature 40 degrees Celsius, which is an optimum temperature. So at this point, this is the max ES complexes formed at this point. So this is the max rate. Why there are the maximum enzyme substrate complexes formed at an optimum temperature? The reason is that if you further increase temperature above 40 degrees Celsius, if you increase this temperature above this 40 degrees Celsius, what happens? The enzymes are globular proteins, and they have tertiary structure. Enzymes are globular proteins, and they have tertiary structure. They have single polypeptide, which is folded and coiled upon itself to make it, this a 3D globular shape. Maybe this has, an active site here. So this may be an active site, and this is 3D globular shape. At very high temperature, what would happen? At temperature above 40 degrees Celsius, at temperature above 40 degrees Celsius, hydrogen bonds present in between these, the tertiary structure. This is the tertiary structure, and there are 4 forces involved. Tertiary structure, which is held by 4 forces. We have discussed them in the biological molecules. There are hydrogen bonding, there are hydrophobic bonding or hydrophobic interactions, there are ionic bonding, and there are disulfide linkages. So there are different forces that hold this polypeptide in a tertiary structure. Hydrogen bonding is also there, which maintains the secondary structure, which is present in this enzyme molecule. So if we increase the temperature, this hydrogen bonding dissociates. Okay, so hydrogen bonding dissociates. So consequently, what will happen? Consequently, the tertiary structure, even the secondary structure, tertiary and secondary structure of enzymes or proteins, that changes. Okay, so as a consequence, if the structure changes, like these hydrogen bonds, okay, these hydrogen bonds from here, they dissociate, so this shape may change into some different shape. So you see, this is, and this shape has been changed. This shape is changed. Initially, there will be less change in this shape, and still substrate molecules can slowly bind into it, and the rate would slow down here. So that is, substrate takes more time to bind into the active site. But at this point, enzyme is completely denatured. Enzyme is denatured, means the shape of its active site, shape of active site is completely changed. Completely changed. So no longer ES complexes are formed. No longer the shape of an active site is complementary to the substrate, so substrate will not make enzyme substrate complexes. So there are zero enzyme substrate complexes. You can say that there are no ES complexes, no ES complexes formed here. Even there are no enzyme substrate complexes formed here at 0 degrees Celsius. Why? At this point, the reason is that enzymes are inactive at 0 degrees Celsius. Like there is very low kinetic energy and molecules are not moving and they are not colliding with each other and they are not making the enzyme substrate complexes at this point. Okay, so enzyme is inactive at this point. But at this 50 degrees Celsius or above 50 or 60 degrees Celsius, whatever is this temperature here given in a graph or given in your question, where the enzyme activity that becomes 0. So this is where no enzyme substrate complexes are formed, as the active site of enzyme is completely changed and it is no longer complementary to the shape of a substrate. So no longer substrate binded to an active site. Okay, so that is how you should explain this graph, and you should explain the effect of temperature on the rate of enzyme action. Now let's see. If we carry out an experiment to see the effect of pH on the rate of an enzyme action, then we should plot pH on x-axis. We may have neutral pH here. We will have, the other values, maybe 1, 2, 3, 4, 5, 6, and 7 here. We will have 8, 9, 10, 11, 12, 13, and 14 here. Okay, so this is the pH range. This is neutral, and this becomes more alkaline, and this becomes more acidic towards this. So this is more acidic. There are different enzymes which are specific at at specific pH. They are most active at a specific pH. For example, pepsin is most active in an acidic pH. Okay, so this is most acidic at pH from 2 to 4. This is most active from pH 2 to 4. And as the pH changes, the rate of enzyme activity that changes. Similarly, trypsin is another protease enzyme, a protein digesting enzyme. Pepsin is found in the stomach, and trypsin is found in intestine, and that is maximum at around 8 to 9 pH, which is acid, which is slightly basic pH. There are some enzymes that are most active at neutral pH, so the graph for them may be drawn like this. Okay, so do you see the enzymes are specific to pH? This is rate on y-axis that we have plotted. So the first point should be enzymes are specific to pH. So as we change the pH, what happens? Why do they change the enzyme activity? As we change pH, why explain the change in enzyme activity? Okay, at specific pH, the enzyme activity is specific. For example, if we look at this graph, the maximum enzyme activity is at this pH. But as you increase the pH or you decrease the pH, enzyme activity changes or enzyme activity decreases. I'll write that point. Enzyme activity is max at specific pH, and change of pH changes rate of an enzyme action. So why does it happen? So as we change pH, change pH, ionic bonds and hydrogen bonds in proteins or in enzymes. You know, the enzymes are made up of proteins. So in enzymes, which are globular proteins or which are made up of, you know, the proteins, and they have a tertiary structure. So these dissociate. These bonds dissociate. So as a consequence of dissociation of these ionic bonds and the hydrogen bonds, the tertiary structure of the proteins or the enzyme, tertiary structure of enzymes changes. So consequently, shape of an active site changes. So if the shape of an active site changes, an active site is no longer complementary to the shape of a substrate, obviously substrate will take longer to bind it to an active site, and this would slow down the rate of an enzyme action. Okay. If the shape of an active site is completely changed or denatured, then obviously substrate will no longer be able to make enzyme substrate complexes, and the rate would, or the enzyme activity would cease, and the rate would become zero. So that's how you should explain the effect of pH on the rate of an enzyme activity. Okay. So let's see some questions on it, and then we'll move on to the effect of substrate concentration on the rate of an enzyme activity or rate of an enzyme action. So let's see. We have a past paper question on this topic, the effect of temperature on the rate of an enzyme action. So here you see figure 2.3 is a graph showing how the activity of alpha amylase extracted from barley seeds changes as the temperature increases from 10 degrees Celsius to 66 degrees Celsius. So initially, you see the rate increases up to this point. So this point has, you know, the max rate at around, you know, you can take precise value from here, but that's in around 50 degrees Celsius. So this is max rate here. So this temperature is an optimum temperature. Okay. So as you increase temperature from 10 degrees Celsius to, you know, as you increase this temperature from 10 degrees Celsius to this 50 degrees Celsius, an optimum temperature, the rate increases from this value. You can take this value like, you know, this box has been divided to 10 units. So there are 1, 2, 3, 4, 5 boxes. So each box is equal to 2 units. Okay, so this is percentage in enzyme activity. So it is 2% enzyme activity at this point, and this is 100% enzyme activity, 100% enzyme activity at this point. So when you increase the temperature from 10 degrees Celsius to 50 degrees Celsius, the rate increases from 2% to 100%. Okay. Then what you should write is, the next thing is, if you increase temperature above 50 degrees Celsius to, you know, here it should be 62, 64, and 66. At 66 degrees Celsius, the rate turns to 0 again. From 50 degrees Celsius to 66 degrees Celsius, the rate slows down, and at 60 degrees Celsius, the rate becomes constant. Here it is increasing, and here it is decreasing. So this is how you should describe it. When you describe this graph, you give two things. Number one, you write relationship between x-axis and y-axis. And you should also give numerical reference. Give numerical reference or the numerical code. The numerical code will be the values that you pick up from x-axis and the values that you pick up from y-axis. So let's see. The question is explain. So explain the effect of temperature on the activity of alpha amylase extracted from barless seeds as shown in figure 2.3, and it is for 3 marks. So you should give a reason why the enzyme activity is increasing from, from this 2% to 100% when the temperature is increased from 10 degrees Celsius to 50 degrees Celsius. And then you should give another reason why the enzyme activity is decreasing when the temperature is increased above 50 degrees Celsius to a 66 degrees Celsius. So let's see. We have already discussed that. So as you see, increase in temperature increases kinetic energy. Okay, so this is one. More kinetic energy here. Okay, so if there is more kinetic energy, there would be more successful collisions. So this would be more successful collisions. And if there are more successful collisions, consequently, there would be more enzyme substrate complexes formed. You will have more enzyme substrate complexes formed. Okay. And that is how that would have higher rate of an enzyme activity. When there are more enzyme substrate complexes, the enzyme would convert more substrate into the products. At 48 degrees Celsius, whatever is, the value from there, 47 or 49, this is an optimum temperature and the rate of enzyme activity is the maximum. Then you see, at a temperature higher than the optimum temperature, denaturation of the enzymes takes place. Why this denaturation of these enzymes takes place? You should give reason for that, and that is that hydrogen bonding that breaks or dissociates, and that leads to changing the tertiary structure of this protein, and that changes the shape of an active site, and that is what is known as denaturation. Hope you have understood it. Now let's see the effect of the graph shows the effect of temperature and pH on an enzyme activity. This is another question. This graph shows the effect of temperature on the rate of enzyme activity, and this shows the effect of pH on the enzyme activity. And this is an MCQ. Which statement is correct explanation of the enzyme activity? We will have to look at each statement one by one. At pH, hydrogen bonds are formed between enzyme and substrate. Okay, so this is a P. And at this point, enzyme substrate complexes are being formed. So if this is, for example, an enzyme, and this is a substrate molecule, so substrate binds into an active site and it is held in its place, maybe with the help of hydrogen bonding. So yes, this statement is correct. So hydrogen bonds are formed in between substrate and an enzyme when the enzyme substrate complexes are formed. So at point P, as there are enzyme substrate complexes formed, so this statement is correct. At Q, the kinetic energy of enzymes and substrate is the highest. This is most challenging part for many of the students. They think that as there is the maximum rate of enzyme action at Q, so it should be the maximum kinetic energy here. It should be the highest kinetic energy here, but that's not the case. Kinetic energy is proportional to the temperature. Okay, where temperature is the max, the kinetic energy will be the max. So temperature is max at this point. This is the max temperature here at this point. So this should be the max kinetic energy at this point. And because this is very high kinetic energy here, so due to very high kinetic energy, the protein molecule cannot maintain or cannot retain its 3D shape. So its 3D shape is changed, and that slows down or that ceases an enzyme activity at this point. Okay, so this is not a correct choice. At R, disulfide bonds in enzymes begin to break. At R, no. If the disulfide bonds in enzymes begin to break, then the enzyme activity should slow down. But at this point, the enzyme activity is the maximum. So after that, maybe there are changes in the structure of a protein or structure of an enzyme. But at this point, R, it is the max enzyme activity, and this is an optimum pH at this point. So at optimum pH, there are no bonds broken down. Okay. And secondly, changes in pH doesn't break down these, disulfide linkages. Disulfide linkages are covalent linkages, and covalent bonds are not simply broken down by changing in the pH. If these substances are boiled in a dilute acid at a very high temperature, then these covalent bonds can break. So this is also not a correct choice. Then we have an at S, the enzyme is completely denatured. So this word completely is not acceptable. That's not the true description of this point in S. Do you see at this point, below this, S point here, the rate of enzyme activity is 0. So at this point, you can say the enzyme is completely denatured. Enzyme is completely denatured at this point. But S is slightly above than that. Do you see this is slightly above, where still there is some enzyme activity taking place. So you cannot say when there is an enzyme activity taking place, even if it is a lower enzyme activity, then you cannot say that enzyme is completely denatured. So this statement is also not correct. So the correct choice will be an A in this case. What is the most appropriate set of controls to use in an investigation into the rate of an enzyme catalyzed reaction over a range of temperature from 25 degrees Celsius to 65 degrees Celsius? Now you need to understand an experimental design and the concept of a control set of control experiment, which is used to, compare your results with. Okay. So let's see an experimental design, concept of an experimental design. Experimental design. So if the enzyme activity has to be investigated at different temperature, we take different temperatures. Okay. We take different temperatures and we place, this is different temperatures at, these test tubes are placed at different temperatures. We put some substrate, this is a substrate put into each test tube. The same substrate is put into, and each test tube, and we put the same enzyme into each test tube. This is same enzyme put into in each test tube. So we are investigating the effect of enzyme activity at different temperatures. And if we have to compare our results, obviously we will see some change in the substrate concentration, and that could be recorded by disappearance of substrate or by appearance of the products, by doing some food test or by doing some using some maybe instruments like colorimeter, where we can measure the changes in product or changes in substrate. So to compare our results, we will have some results obtained at the different temperatures. We take another test tube, okay, which has the same substrate placed into it. But instead of this enzyme, we put either the same volume of water into it, we can put water into it, or we can put boiled enzyme into it. So there is no enzyme present in this test tube, and you will see uh, no change in the substrate concentration will take place here. Neither the product will be made nor the substrate will disappear. So the results of this enzyme activity could be compared with this. So this is known as control set, or this is known as control. Okay. So this is what we have to find in this case. So let's see. The question is, what is the most appropriate set of controls to use an investigation into the rate of an enzyme catalyzed reaction over a range of temperature from 25 degrees Celsius to 65 degrees Celsius? Enzyme and substrate are all enzyme and substrate at all temperatures tested. This is an investigation, not a control. Enzyme and boiled substrate at all temperature tested. So we don't need to boil a substrate. We are investigating the effect of enzyme. So enzyme should be boiled. Okay. And in this case, boiled enzyme only at all temperature tested. So enzyme plus substrate must have been added into it. All other conditions have to be constant. So this is also not a correct choice. Okay, this is also not a correct choice. And here we have substrate only at all temperatures tested. So no enzyme added into it. So this investigation or this experiment has enzyme catalyzed reaction, enzyme added into a substrate, and this has no enzyme added into it. So we can compare the results together. So this would serve as a control experiment. We can have no enzyme added into it. We can add same volume of water into it, and we can have boiled enzyme plus a substrate added into it. So these are different ways we can set a control experiment. But substrate should be added in each case so that the rest of the other conditions except enzymes are present in every experiment. So D is a correct choice in this case. Here we have the effect of temperature again. The graph shows the effect of temperature on the rate at which enzyme in a biological washing powder digests and remove the fruit juice stains. Which statement explains the shape of graph at temperature higher than X? Okay, so we have to just explain this part of the graph, not the first part of the graph. So do you see this rate of enzyme activity? Rate is decreasing in this case as this temperature increases. So the question is, which statement explains? So what is happening is, as this temperature increases, the enzyme activity decreases. Why? As enzyme denatures. Okay. So as the temperature increases, the denaturation increases, and at this point, it is completely denatured. At this point, it has started denaturing, or it has started dissociating hydrogen bonds, and as a consequence, the tertiary structure and even the secondary structure of an enzyme or a protein is changed. So shape of an active site changes, and consequently, there are less enzyme substrate complexes formed. So let's see. We have bonds are broken down between the R groups of the amino acids in the polypeptide chains of the enzymes. Yes, maybe when you increase the temperature. So let's see this. We'll discuss this point later. So there are more collisions between the enzymes and its substrate. Okay. So as you increase temperature, the kinetic energy increases. There may be more collisions in between them, but these are not the effective collisions. So if there are more collisions taking place, then the enzyme activity should not slow down. So this does not explain this actually, the graph. The rate is slowing down. And if there are more collisions taking place, then the rate should increase. So this statement does not explain it. The tertiary structure of enzyme is altered. Yes, this statement explains it. And the shape of the active site and the substrate are no longer complementary. Yes, this also explains it. So you see, we have 3 and 4. We are sure that these two statements are correct and 2 is not correct. So as 2 is present here, so we reject this. 2 is present here, we reject this. And 2 is also there, so we reject this as one. So only the choice left is C. So it means that bonds between the R groups of the amino acids and the polypeptide chains of the enzymes are broken. As you increase the temperature, obviously the bonds in between the R groups that may be changed or broken. So consequently, this statement is also correct. So C is a correct choice in this case. Okay. So this is another question here. This is the last question today. The graph shows how the concentration of components 1, 2, 3, and 4 of the enzyme catalyzed reactions change with the time. So over a period of time, what happens? See, if we look at it one by one, so this is time. Over a period of time, what would happen? Substrate will be converted into the product. So substrate concentration that would decrease. Okay, so this should be substrate. Okay, so this is concentration of substrate. Over a period of time, as substrate is converted into the product, so substrate concentration will decrease, but the product concentration will increase as substrate is converting into the product. So product concentration should increase. Okay, so this should be a product. Okay, so let's see. 1 is product and 3 is a substrate. So this is 3, component 3 is a substrate. So this could be this one or this, and 1 is a product. So this could be this or this. And what is then 2? 2 is, first slightly decreasing and then becoming same or the constant. So it should be an unbound enzyme actually. And this is, initially the amount increasing and then becoming lesser. So enzyme substrate complexes would, in the beginning, would be more made. And as the time passes, the enzyme substrate complexes will decrease. So that should be, so D should be the correct choice in this case. So that's all about the effect of temperature and effect of pH on enzyme activity. In the next class tomorrow, we will discuss the effect of substrate concentration and enzyme concentration on the rate of enzyme activity. So that's it for now. See you tomorrow, inshallah.